In this video, we'll start talking about nervous system, and let's really focus in on neurons. So you know from past courses that a neuron is an excitable cell, and it's about receiving and transmitting or sending information in terms of action potentials. And most neurons, we're going to see there's quite a diversity of neurons, but most neurons have, of course, the cell body or soma, and it's metabolically active. This is where we make all the proteins, everything that's needed. Neurotransmitters are made up here. We also have endoplasmic reticulum because, of course, there's protein synthesis, there's rough ER, and the rough ER actually we'll give a new name. We'll call it Nissl bodies. Golgi apparatus, usually there's a large or multiple nuclei, and of course there's mitochondria that we typically see in the cell body of a neuron. Now, the cell body also tends to have an extensive cytoskeleton. So we're looking at microtubules that give support and help with chemical transportation between the cell body and the axon. There's neurofibrils, and these are intermediate filaments. In past chapters, we've talked about some of these filaments, and they give structural support of the different parts of the neuron. Now, we also have the dendrites, and depending on what type of neuron you're looking at, they have different appearances. But typically they're short branched processes, and really typically they receive information from other neurons. And information is transmitted from the dendrites to the cell bodies. Now, each neuron can have multiple dendrites. And as I said, there's multiple shapes for different categories of neurons, but we'll have a look at that shortly. Also, when I'm looking at other parts of the neuron, we're going to see some terms. Now, we have, typically each neuron has one axon, and we can call that the nerve fiber. And of course, this is where we generate and conduct action potentials. Now, at the start of an axon, it's really where the cell body starts to taper down and become the axon. There's a region called the axon hillock. Now, this is going to be an important region when we start talking about the formation of action potentials in the near future. You can actually have axon collaterals. So these are the branches that may extend off an axon. Not every axon has these extending branches. The telodendrita, they're small branches arising from the axon and the axon collaterals. And we also have the axon terminals or synaptic bulbs. And this is really where we have the communication between one neuron and the next, and we'll have a look at that. The axolemma, now look at the last bit, lemma, gives a suggestion when maybe we're talking about plasma membrane, and indeed it is the name we give the plasma membrane. And the axoplasm is the cytoplasm within. Now, just a little FYI is that, of course, the axons, things are often transported down them and back up the axons. So we have to have axonal transport or flow. And there's two main types. We have a slow form, which we call slow axonal transport. And this transports substances, things like cytoskeleton proteins and a few other things. And this is a slow way of traveling from the cell body through the axon. And it's about 1 to 3 millimeters per day. Now remember, there are some axons that are quite long in the body, the ones that run down through the spinal cord or in the peripheral nervous system. So 1 to 3 millimeters per day is not that far. We also, in terms of, isn't a whole lot of distance in a given day for that transport to occur. The fast axonal transport, this requires motor proteins, and it consumes ATP in order to move stuff down very quickly. And these are typically vesicles, membrane-bound organelles that travel back and forth. Now, there's two directions, of course, going from the cell body to the synaptic knob. We call that anterograde. And things that flow from the synaptic knob back to the cell body we're going to call retrograde. And in this type of fast axonal transport, we're looking at somewhere between 200 and 400 millimeters per day. Now, that's quite a difference between that 200 to 400 millimeters per day in fast axonal transport. And just reflect back, a slow axonal transport is only 1 to 3 millimeters per day, so it's quite a difference. Here's just a picture. Here we can see the cell bodies. We can see different processes, some axons, dendrites coming in, etc. And of course, these dark little cells, these are the neuroglia, or also known as glial cells. You can call them each one. You can use each of those names if you wish. Let's have a look. Here's a typical looking axon. And here's the cell body. We have the dendrites, multiple dendrites, right? Nucleus, Niesel bodies, which are ribosomes and the rough ER, mitochondria, the nucleus, intermediate filaments that give it the shape, and etc., neurofibrils. And here's the area that is the axon hillock. So it's this area right here where the cell body really starts to narrow down, and it really is right at the initial segment of the axon. Many axons just run straight. Some of them do have branches, which we call collaterals. Right? And we'll come back and talk about the myelin sheaths in later videos. Now, just a little FYI is that it's interesting. Some viruses actually use our retrograde axonal transport as part of their infective process to get to the cell body. And one classic example is polio. Now, polio is being mostly eradicated from much of the world because of effective vaccination. There are still some parts of the world where polio is still endemic. And it's caused by the polio virus, and in the infection impacts the central nervous system, especially the spinal cord, and that's why you tend to see paralysis, the weakening, the shriveling of muscles as the muscles become paralyzed because of loss of motor input into them. And just a little FYI, polio until up until the 1950s was really sort of the scourge of pediatrics, because children would be healthy one day and then a week later they could actually be paralyzed lying in bed, and many of them never recovered from it. So that was one of the greatest triumphs of vaccines. Now, no cure exists, but polio can easily be prevented by vaccination. And what happens is the virus actually accesses the central nervous system by entering cells. It could be a muscle cell, etc. And it passes into the motor neurons at the neuromuscular junction. And it travels the length of the axon by retrograde transport until it reaches the spinal cord. And there, once it enters cell bodies, it can start the division process and viral replication. Now, polio is not the only virus that does this. The group of viruses that fall under the herpes family of viruses, and it's quite a diverse family of viruses. We tend to think of like cold sores as that's herpes, but there's a number of other herpes viruses. Rabies is another classic virus that uses this, and some toxins have the ability to invade neurons by this method too, like tetanus, the tetanus toxin, which causes, of course, tetanus. Now, we're going to wrap it up there, and we're going to come back and start talking about the shape of neurons, the regions of neurons, and looking at sort of the diversity of the shape and function of neurons.